then the lab is not going to go as smoothly for you. So everything that came from here should be in your notes. Okay, a couple places you can look. We had the first notes, the first day notes, where you could, I guess the second day, where we listed the different biomolecules, and then we listed the functional groups here. So you also have structures of functional groups here. Then you have how the organic molecules bonded together. And then you had a chart that you so far have filled in this column, and that's it. You've drawn sketches of all these guys. So you should have for each of these functional group biomolecules, you should have a functional group structure and a name in your notes. You should have the biomolecule name and structure right here. And in this, you should have circled the functional group that's important. How are we feeling about that so far? Okay. So that information from those pieces of your notes and from that chart will let you fill this in. How are we doing with this? You should have this pretty well completed, either the hard copy or the copy in classroom, either one. I don't care which one you do, but whichever one you do, you bring it in when you're done. Later that you're finished, you might take a look at your stuff. So how would we start out the list on the left? Somebody share what you have in the list on the left. Got that? Oh, go ahead.
That's the phosphate, ring, the N8, 3 plus H2, and COOH and OH. Right.
You guys say. Are you okay with that? Good, good, very good. Okay, then we're going to do the corresponding organic molecules or biomolecules on the right. He's got those. There we go. Okay, good, good. So if you guys have this, if you can correlate a phosphate group with an ATP and a carbon nitrogen ring with a nucleic acid, you're pretty good. Okay, that's pretty good. Then this chart is just kind of an extension of that, where you're going to put the structure. So here you're going to draw the picture of the functional group, which is right in your notes. You have those in that one page of notes that we looked at. You're going to draw those. Then you're going to name them. Then you're going to name the biomolecule they identify. So again, you're going right back up here, and you're putting 1 and 1 down here, name and name, structure here, and then you're going to draw the picture here. Where are you going to get those pictures? Where are you going to, Levi, we're going to get them? You don't want to do resource, yes, there's one place where else. On that chart. Remember the day you took the pictures of all this stuff, you also drew them on, this chart in that first column. And that's all you've done so far. That's all you've done is draw the pictures in here and circle the functional groups. All right, how are we feeling about that so far? I got it. I have questions. I don't know what charts you're talking about.
The basic thing makes it what it is.
So for ATP, what would you?
The 4 O's and 2 S's.
Yes, phosphate group. That's right. Good. Okay. You're all good with that? Awesome. And then you're going to write yourself a little paragraph or a set of steps or directions of what you're going to do with that. So just like it's like Kyra said, you're going to write down, well, I'm going to look for the phosphate first to identify an ATP. If I find that, then I'm done identifying that molecule because none of the others have phosphate. So it's going to be your writing your directions. Now, here is the whole reason you're doing this. So that you could look at a molecule and you could know from its structure what kind of molecule it is. Now, you don't have to know that this is a glucose or this is a valine, okay? Or that's a cytosine. You don't have to know that. You just have to know, well, it's one of 20 amino acids. So what you want to be able to do here is this. In the directions, you're told to circle all the functional groups. So here, we found the functional group, we found the NH2 is a functional group, and we found the carbon-nitrogen rings are functional groups. We circled them. Which one of those will let you identify this molecule? Go down the order. Which one? Yeah, because you're going to go in order. You look for phosphate first, then a carbon-nitrogen ring, then an amine group. So the CN ring will let you identify that. So you do a box around the CN ring. So that works? All right. Did anyone get through this whole page and think you did pretty well? Who get through this page and think you did pretty well? You kind of felt pretty good about what you were doing? Okay, what questions do you have? What questions? Kyra, what do you got? This one? Okay, why was that confusing for you? Talk to me.
I was since it had the H3 and the ring. I don't know. What if it had both?
Oh, good catch. So what all did you circle here? You can circle this. What else? This, okay. Because that really is a double bond there. And this, you've got 3 things you could circle. Now, when you're looking through the steps, we didn't have what? Which group is not here? The phosphate. You didn't see phosphate, so you can eliminate ATP. But now you've got a CN ring, you've got an amine group, and you've got a carboxyl group. So the rule would tell you that you go with this, right? Now, look at the others that are carbon-nitrogen rings that you have. And the carbon-nitrogen ring is pretty much central when you're looking for that. Central here, and it's central down here. Okay, they're central. Is the carbon-nitrogen ring central in this guy? No, it's kind of stuck on the edge. What it is here, it's actually an R group. Remember the R groups from your inquiry that you did when you talked together? Yes. So here, you'll say, well, it's really not that. What comes after the CN ring? What do you look for after that? Sure, do you have one of those? Bam, and that's what you're going with here. Okay. Because the CN ring needs to be central. It needs to be the main part. And here it's really just an R group. Any of the others that you want to use? Any of the others? having gone through this much, then I'm going to give you a couple of minutes to rethink and look again at what you did and make sure you're happy with all the choices that you made. And we can talk about any of those that you want to talk about. So I'll give you a couple of minutes there to play with that. Anything else you wanted to talk about or think about or check to see if you did correctly or talk with a friend? Remember, you're circling all the groups. You're going to put a box around the one that lets you identify what this is. So here, in the center one there that we discussed, what are we going to box? We're gonna what? The reason that you did this back page is because these are the main big things you need to be able to do with this. We're going to be able to look at one of those molecules when we see it during photosynthesis or cell respiration, or when we're building cell membranes, or when we're making proteins and protein synthesis. And protein synthesis, you're going to use two of these guys. You've got to be able to tell one from the other. So just this quick look at, well, do I have a carbon nitrogen ring, or does it look like an amino acid? Just a quick ability to make that determination is going to make learning those things so much easier for you, and you can do that now. Anything that you want to talk about? How are you feeling about this now?
Yeah.
Better? Good. What question would you still ask? What would be helpful?
I think the circle is quite okay. Well, the sharp one for me,
Is helpful, or you need help with it?
like that's probably the most difficult part for me.
Okay.
Figuring out which different ones to circle. One different.
So, is it helpful then if you go back over to your chart and you look for the structures that you made here? So you look over here like for this, or you look for this. So remember to look for them or finding them. Okay, so just remembering the order that you go. So it's sometimes helpful to think that first you look for P, then you look for N's. And those are the first three. After that, you're looking for COOH or just OH. You just have to get that in your head. Anything else about these? I think we're going to look at a little quiz on these in a few days. But first, we have 2 days of your lab to work through, where you're going to be testing different substances to see if they have in them, if they have amino acids for proteins, if they have lipids for fats, if they have either starch or a sugar, which would be carbohydrates. We're going to be testing some stuff for those. And so here's how you're going to do that. If you would go into classroom and pull out this lab, which is new today. And what you want to do with this, because you're going to be working on it multiple days, and I don't know how to actually answer that, you may want to just save a copy right now in your folder, in your add files folder. So this is linked to today. Also, if you're looking at your grades in classroom, don't look at them in progress book. If you see anything that doesn't look right, let me know. Because some of them sit in classroom and some of them don't. And sometimes that messes up and sometimes I mess up. So let me know if something doesn't look right. So let's go here, look at what you're doing, and your introduction part is due tomorrow. So we'll go through this and I'll let you have some time to get started on that. So here's what's going to happen in this lab. Are you there yet? Are you all saved? Is in your folder? So neither of us has to go looking for it? Okay, good. So what we're going to do here is we're going to be testing different substances. We're going to do this in two parts. The first part, you're going to test substances that you know what they have in them. Those are called standards. So you're going to have a solution that you know has protein in it. You're going to have one that you know has starch, one that you know has sugar, one that you know has fat. Why would we do that? Why would we do that? Sure. You know exactly how something's going to look if it has protein in it. So you know what they're going to look like. So you'll test those tomorrow. I'll let you take pictures of them if you need to, because we won't be able to save them. And then on Thursday, you're going to do part 2, where you're going to get some substances that you don't know what's in them. You're going to determine that. And you're going to use real life substances that come from the real world. So in your introduction, you always do some preparation for lab so that you know what you're doing. And here's what you're going to do. You're going to write 4 paragraphs, roughly a paragraph for each of the bullet points. You might need more, and some of them might be a short paragraph. In the first paragraph, you're going to say the 4 types of biomolecules are, and you're going to talk a little bit about each one. So you're going to say, one type is an amino acid, and it makes up proteins, and here's what proteins do. So you might need to do a little bit of Googling around for that. Don't go into a lot of detail. Don't write a bunch of crap that you don't know what it means. It's pretty simple, but it just needs to give an idea of what those things are. So your first paragraph is going to be just listing the 4 types of biomolecules and telling a little bit about them. Okay, then the second paragraph, why do we need to eat those things, or why are they part of our body? What do we need lipids for? What does it do? And again, you don't need a dissertation on the chemistry of fats. You just need to say, this is why we eat them. This is what they do for us. It can be short and sweet, but you need it. Now, the how you'll test for each. We've got a procedure down below. I wrote that for you. You're welcome. Okay. And I'm going to walk you through that and show you what you're going to do. So you're just going to say, well, here's how I'm going to test for each. To look for protein, I'm going to add some Bio-Rad, and if it turns purple, protein's present. Easy enough? I'm going to show you those right now. And then what's the purpose of using standards? Why are we using the ones that we know what's in them? So here's what you're going to do. As you're working or as you're reading the procedure, you can do the materials and equipment tonight too. List everything you're going to use. I made you 2 columns to put those in. That little text box there in the table will expand. Safety precautions. You will need to wear closed-toed shoes. We will wear chemical splash goggles, which will get sanitized and covered between uses, as usual. So here are your tests. You have a test for sugar, a test for starch, a test for protein, And the lipid test we may change because I haven't found this to Dan yet. It was in the refrigerator, but it's not now. Okay. So Benedict's test for sugar, here's what you're going to do. It's really important that you read labels carefully because here are two of your test solutions. They both start with B and they're both blue. So you need to read carefully. So the Benedict's is this one. And you'll have 5 test tubes labeled A, B, C, D, and E. So in each of these, you'll put a different solution that you're going to test. So A might be protein, B might be sugar. You're going to put those in here. You're going to test them all with Benedict's. Got that? You're going to see what they do with Benedict's. One of them will turn color. The sugar, and you might want to write down. I'll wait while you get your notes. You'll remember to find it. Write the last left off your notes so that you know what they're going to look like. You'll expect that Benedict's starts out blue. It will turn any color but blue with sugar when you heat it. Benedict's turns any color but blue when you heat it. Benedict's turns any color but blue with sugar when you heat it. This is the only one you have to heat. I'll show you how to do that. So you've got your 5 test tubes. You're going to put a different solution in each of these. They're labeled A, B, C, D, and E. Your solutions will be labeled A, B, C, D, and E, but they're going to be on your table in little paper cups. So you're going to put in 5 drops of Benedict's. You're going to have put in 5 drops of solution A. Then you're going to take your test tube rack. There will be 2 hot plates at the front of the room that will let you heat. 1 person per hot plate. They'll be at different ends of the table so you can socially distance. And you're going to work, by the way, with the same partner that you worked with before. You're going to be one pair of you at each of the tables, and you're going to be as distant from each other while you're working as you can. So you're not going to be huddled together. You're going to be apart. So you'll bring them up here, and then this water will be hot, but it's not like boiling, scalding hot, but it's hot. So you will use your test tube clamp, and you will place your test tubes in the water. You can just kind of drop them all in there and leave them. Right? You'll leave them for 2 or 3 minutes. You will see if they're going to change color, they will do so within a minute or 2. And you'll see anywhere from a greeny color to anywhere orange and brown. The more sugar, the more towards orange it gets. So any color change from the blue is going to show you that there's sugar present. Okay. That's the hardest test there is to do with that. You're all going to set this up first. And then you go up and get this done whenever there's space at one of the hot plates. I might just set up a 3rd hot plate. So it's your Benedict's. You're going to bring them back to your table. You're going to dump them, wash them out really good. You're going to refill them with solutions A, B, C, D, and E. And then you're going to use Rugel's iodine. So this is the iodine test. Iodine, when starch is present, iodine turns black. Okay, so you can write down iodine turns black with starch. And for this, all you have to do is add the iodine to your solutions in your tubes. You don't have to keep this. Just going to put the iodine in, and then you can kind of shake it around a little bit, and you'll see it'll either turn black or it will stay tan. Tan, no starch, it's black, that's starch. You're going to then wash out your tubes. You're going to do the same thing. Fill each one, solution A, B, C, D, and E. You're then going to use the Biomolecule, which will turn a really lovely shade of purple if there's any protein present. Purple protein, good. So Biomolecule will turn purple if there's protein. And the Sudan, if I can find it, it was in the refrigerator, it's not now. The Sudan is red, and if there's oil, it will absorb the red and kind of pull it from the solution and separate out. An alternative to that is to put some of the substance on a piece of paper towel and see if it leaves a grease spot. Big piece of ox. Leave a grease spot, big piece of ox. Yeah, now y'all know what I'm talking about, don't you? Is anything real complex there? For the standards for the first day, you're going to just record your observations in the status table. So in the first column, you'll mark down what color everything turns with Benedict's. Now, if it turns, if you put the Benedict's in, it looks blue. If you heat it, it doesn't change, you're going to put down blue. Okay? You're not going to put down an X or an MA. You're going to put exactly what you saw. If it turns color, then you're going to note that. So this will give you a visual of what you just wrote down. If you would like to take pictures of all the standard sets, you may do that as well. Just let me know if that's what you're doing and you're not going to Snapchat your friends. I don't get it. People can Snapchat their little hearts up. They can't get friends. Anyway. I can make a web page on my website faster than I can get traveling faster. But that's either here or there. So that's what you're going to do. The next day, on Wednesday, you're going to have 5 more samples. You're not going to know what they are. Right? They're going to be probably some kind of food. And you're going to test them to see what they have in them. So it's very possible if you had, for example, a milkshake, you might find any number of things. It might have any number of these things. It might have 1, 2, 3, or all of them, or none. That's kind of scary, isn't it? Why did I eat that? And then so you can do the same thing with the unknowns. Now, after the first day, or even pretty much now, you'll be able to go through some of these analysis questions. You can answer what color things are supposed to turn. You might have to wait till tomorrow. You can do those. The conclusion is going to be plain evidence reasoning again, just like your pH one. It should be a lot easier because you're going to look at your results at your chart. And you're going to say, well, this food had this, and this biomolecule. This food has this, and this, and this biomolecule. Or you could say protein was found in this, and this. But that's all you're doing. Food, what biomolecule? And then in evidence, you're going to say, this food turned purple with Bio-Rad, this food turned black with iodine. Okay? And again, you can group them. You can say these foods had carbohydrates and turned this color with iodine. Down here, you say the reason. You say, the egg contained protein. We know the egg contained protein because it turned purple with the Bio-Rad. Then you're going to talk about the limitations and the other issues that you had with this. That That conclusion writing will be your job on Friday. Okay? And this whole thing will be due on Monday. The introduction is due tomorrow when you come in. And so 4 paragraphs that you have to write. But you don't really have to think of anything because you know all the stuff, except for a few things you might want to look up. Okay? To get questions. How much time do we have left? Oh. So that's enough time to get a good start on that introduction. So, if you have questions, I'll walk you through any part of that that you have a question on.